So good evening, good morning, colleagues. Thank you for joining us on this webinar that's being hosted by the University of Witwatersrand. And it truly gives me great pleasure to welcome back, in a sense, an alumni of the University of Witwatersrand, Dr. Patrick Soon-Shiong. So when I was reading through his CV earlier today, or at least his bio sketch, I figured that it would be a large waste of our time if I were to go into the numerous achievements of Dr. Patrick, except to say I think the single words to describe him pretty much paraphrases the breadth of his contribution to the field of medicine, his contribution to the field of science, his contribution to the field of research. So Patrick is a physician, a surgeon, a scientist, an inventor, an innovator, a technologist, a philanthropist. He's got a number of firsts, including a number of firsts when it comes to cancer therapies, a number of firsts when it comes to innovation in the field of surgery. And for me, it was a real pleasure just to have been introduced to him just a few months ago. And over the past perhaps 8 to 12 weeks, I've really gotten to know more about his work and really the passion that drives him to really look beyond the boundaries where many other scientists fear to tread. And when it comes to COVID-19 vaccines, which is his latest initiative in terms of looking beyond what most of the other scientific communities are looking at when it comes to developing vaccines, which really would be, in a sense, the next generation of COVID-19 vaccine, he certainly doesn't fall short. So Patrick, I'm not going to waste time. I know that our time with you is fairly limited. And again, a hearty welcome back to your university where you graduated in 1975. And were it not for COVID-19, we would have hoped to have this in person. But we really look forward to welcoming you in the near future, and perhaps next year when we celebrate the centenary of the University of Witwatersrand, that will be a golden opportunity for us to again host you in person. So thanks for making the time, and look forward to you presenting to us your thoughts on COVID-19 vaccines and what you see to be the future of COVID-19 vaccines, and how South Africa as a country, and how perhaps Africa as a continent, which is where your roots really exist, should move forward in terms of taking on the initiatives that we can become self-sufficient. So thanks for making the time, and over to you.
Shabir, thank you. What a pleasure this is the first talk again to Wits that I've had now since I've left the country. So it's an amazing pleasure and honor to give given the opportunity. And, you know, I think credit to Wits and the giants in which we sit on, that we're willing to not only take chances, but actually go where other people don't want to tread. And we have, as you know, I've trained under Professor Du Plessis, Professor Myberg, Professor Seftel, Professor Bothwell, Barlow, and each of those have contributed to the science of the world. And I'm so proud to see that Wits and South Africa is now contributing, I think, leading the science for COVID. So Shabir, I'd be happy to just give you a few slides and then have a chat or take questions, whatever is most convenient. And I've got time, so it's important for me to sort of share how we see the world of COVID, very much like we see the world of cancer.
So Patrick, I think it would be great for you to start off with a brief presentation and overview in terms of how you see COVID. And then for the participants, if you do have any questions, I ask that you just post it on a Q&A, and then I'll forward those to Patrick. And then we can have a discussion, not just around the presentation, but just generally around COVID. And Patrick, I mean, it would be good, you've already started to share some of your experiences a bit. It will also be good for you to provide some guidance. We've got some students also that are attending this evening, perhaps to have some words to them as well. But we can start off with you presenting, and then we can take the discussion as it evolves. Thanks.
Well, it's my greatest pleasure when there's students around, because I think that's our most important contribution to this next generation, pass on whatever we think we know. So let me start first about how we designed this vaccine, and maybe Phil, slide 7. I'm sorry, I'm just going to run through my slides in the short form. And I think the rational design of a vaccine we've taken from the perspective of cancer. So for the last 5 years, I've devoted our efforts to seeing how could we induce tumor-associated antigens and natural killer cells and T cells. And we really believe that the T cell was important. So therefore, if you look at A, we had a vaccine platform that can induce what we call PAMSODAMs, meaning we could induce the activation of the patient's own immune system. And if you could deliver a sequence repeatedly, even in the presence of pre-existing immunity, and this is why we identified on the right-hand side a adenovirus that can be given in the presence of pre-existing immunity. The next question is, how do you select the antigens? And it was very apparent to us, very much like cancer, that mutations is inevitable. In cancer, giving targeted therapy, unfortunately, is a little bit futile because the cancer's constant is ability to mutate. Very much COVID's constant. You give it antibodies, it will mutate around it. And therefore, it's not surprising what you're seeing in South Africa, the 484 mutations, which then said we needed to go not beyond just S, but into the nucleic capsid. And surprisingly, we're one of the few that have taken this approach of S plus N. But when we started looking at N, which is an intracellular protein, we discovered, and together with work that is done by the NIH and Dr. Neal, who showed that the nucleic capsid protein of this SARS-CoV is unique in the sense that it uses the lysosomes to escape. But we want the lysosomes to actually break down the protein so it can have CD4, CD8 T cells. And so therefore we generated a sequence that would actually drive it to these lysosomes. And therefore we now have a very potent CD4, CD8 T cell activated together with the antibodies. But we also realized that it's important for you to have cold chain and supply. As you know, because I grew up in South Africa, -80 freezers and all the way in Africa and poor developing countries, there's no way we could have a distribution. And vaccination requires distribution to people, not only we could afford it, but access it, which meant then we needed to figure out the impossible task of generating this adenovirus in a thermally stable capsule. So the idea being, however, that subcutaneous, intranasal, inhalational, sublingual, next slide, 8, 9, Phil, would be an opportunity. Sorry, I hope I'm not messing up your board here. Let me just clean this up. An opportunity to take one vaccine, multiple routes of the same vaccine to achieve both T-cell, antibody, and mucosal immunity. And therefore, we started with the subcutaneous form on the left-hand side, and that's in clinical trials now in South Africa. By April 2020, we had a full GMP and were allowed by the FDA. Unfortunately, there was a long delay by the FDA to allow us through the gate. And even though we had full GMP by April 2020, we received the approval to initiate in October 2020. But by that time, by August 2020, we had already developed this oral capsule. And by December 2020, we've begun and now have a sublingual tablet. It is remarkable that you could have one vaccine designed across SNN. And I'll take slide 10 to really walk you through what I believe is the future. Not just for COVID, but for influenza, TB, leprosy, Lassa fever, chikungunya. So this is the opportunity for us to take this technology now and expand it for countries that really need these kinds of vaccinology. So you will see here, we've taken a COVID-19 SNN construct, but what I call SNN ETSD construct. We've taken then and inserted that into a second generation adenovirus that can be given in the presence of ad immunity. It's a what we call E2B deleted adenovirus. We've then developed a proprietary nanoparticle molecule that would protect this adenovirus from the acidic pH within the capsule itself. We've then brought in a proprietary molecule from Bios that they've worked on to maintain the H85 at greater than 45 degrees centigrade. We've then invented an enteric coating for release inside the lower GI tract. So this capsule required multiple layers of innovation to achieve its goal. And now on the right-hand side, you see we've now scaled it, and what you see in that is the world's first capsules that we made. And in fact, the patients have been dosed as of last week in the United States. The goal in slide 11 is to have what I call a disruption. I had a call yesterday with the World Health Organization, and it was very inspiring because I think the World Health Organization felt this was what they call constructive disruption, and not that by oral delivery, a potential of self-administration, no healthcare worker required, no needles, no plastic, no vials, with repeat dosing, which we've shown, in which the vector could be used not only in cancer, which we've now shown in about 13 phase 1/2 trials with the National Cancer Institute, and where we've taken colon cancer, prostate cancer, multiple solid tumors, pancreatic cancer with this technology. But now we can repeat dose, and we've shown this repeat dosing in our non-human primates, which I will share with you. The concept of quad immunity, meaning that not only would we have antibody and T cell, but memory T cell and memory B cell and mucosal and IgA and systemic immunity. In that instance, we would have, we believe, long-lasting immunity, and we know that to the end you have long-lasting immunity. Thermally stable for global distribution, and most importantly on the right-hand side, specifically for the world, antigenic drift. We saw right from the outset, antigenic drift was such a predictable event that it was futile for us to treat this like flu when there's 7 billion people involved with this needing this vaccine. I will show very quickly the results on the non-human primate study, and because of time, and I'll take you to slide 20, in which this was about a warp speed sponsored study in which we gave sub-Q and oral, and from days 0, 14, 28, and the animals that were then vaccinated and then transferred to a BSL-3 facility and then challenged. And I think what was really exciting is this challenge is a significant challenge, and then daily measurements of the viral load and viral replication. And the goal would be that if a sub-Q oral formulation could inhibit not just replication and transmission as well as disease, but now there's an opportunity to show that we can inhibit replication and clear the virus from the outset. So during this phase of this immunization, we had blood samples taken along here for both T cells and for antibodies. And let me show you the T cell data on the next slide during the vaccination. And here you see very specific S1 and N T cells. And as you could see, very high levels. And these are gamma interferon levels by day 14 and day 35 of T cell activity, which was Th1 dominant because there was no IL-4 at all. And the ratio of the dominance was very high. both on day 14 and day 35. So this formulation of sub-Q oral and oral, when we looked at the data after the challenge on slide 22, what was very exciting and interesting is evidence that we have now memory B cells. So I think it's illusory. I'll be careful about just measuring antibodies in the presence of memory B cells. So what happened on the day of the challenge, huge potent release in all the animals of neutralizing antibodies. Immediately, this is close to 1 to 5,000 to 1 to 7,000 dilution. Excitingly, on slide 24, we then saw, if you put this together, the T cell data, which is Th1 dominant, the memory B cell, and if you add these two together, what we saw was the immediate drop in replication of virus in the nose, immediate drop of the replication of the virus in the lungs, and the viral load immediately dropped to undetectable levels. This study was performed not in our hands. All we did was provide the sub-Q oral and oral. So we are very confident that we had then a system on slide 25 of a second generation vaccine that would generate not just antibodies, but more importantly, T cells and provide protection. So we believe that antibodies plus T cells would allow rapid clearance of SARS-CoV-2 from the nasal and airway tissue. What's torturing me is the fact that we are in the process now of trying to convince the world of vaccinology that we should look at T cells as the dominant form of protection. While antibodies are important, T cells matter because they kill and clear. So on that basis, slide 27, we've taken the approach to say we are now launching in the USA the first two cohorts, the first phase 1, sub-Q. But we then needed to go to sub-Q sublingual, and we've now launched sub-Q oral, and now in South Africa, sub-Q oral and sub-Q. Where do we want to go from here, and where should we be going from here? Slide 28 is to really look at taking one vaccine, 3 routes of protection, and actually looking at the potential to be the universal boost for all vaccines, whether we the prime or anybody else is the prime, is to take our vaccine and boost a Johnson & Johnson vaccine, an AstraZeneca vaccine, Moderna vaccine, a Pfizer vaccine, with the concept that you have antibodies, we bring T cells. And if we could boost with the oral capsule, that would be even ideal. And then the idea is to rapidly run into a standard phase 3 trial against placebo. Unfortunately, I think today it's difficult to take a placebo patient. I think better for us to take all patients that are either infected or had a vaccine and demonstrate that we can generate what we call enhanced immunity through T cell generation. So Shabir, this is a shortened version of my presentation. I'm respectful of the fact that there may be not enough time with regard to people wanting to ask questions, and I'm happy to take as many questions as you like.
Great, Patrick. So I've been sort of monitoring the Q&A, and it's meant to be questions, but it's just compliments about the amazing and stunning work that you're doing. And I think when you call this constructive disruption, I see it not as being disruption, but as creating havoc in the field of vaccines. I mean, you sort of treading where what has really been the holy grails of vaccines in terms of, especially from low middle income countries when it comes to delivery. I mean, we've got the intranasal vaccine, which never really took traction because of the storage requirements, even with the intranasal flu vaccine as an example. But when you start talking of sublingual vaccines and oral vaccines, enteric vaccines, it obviously is a completely different ball game. So really, really interesting. So Patrick, like I said, for the audience, if you do have any Q&As, please feel free to put it up, and I'll pose it to Patrick. And Patrick, while I'm at it, Cheryl Levitt was in your class, conveys their regards as well. Okay, so how long does it, one of the questions that's come through, how long does it take to produce T cells post-vaccination?
Sorry, Shabir, what was that question? I didn't quite hear.
How long does it take for the T cell responses to be induced?
Yeah, no, good question. That's what's the beauty about T cells. It's induced very rapidly. We've even seen it as rapidly as 14 days, but by 21 days, you have potent T cells. So actually from the prime alone, we have T cell induction. Antibodies take a longer time, sometimes 37, 40 days, 50 days, but T cells, N and S specific T cells, and specifically N T cells, they are induced very rapidly.
Okay. So Patrick, let's just deal with this a little bit more. So you're obviously making an extremely strong case for T cell immunity, and I certainly probably do believe that T cell immunity plays an extremely important role in terms of protection, especially from severe disease. The question obviously is, if you're wanting a vaccine to bring about an interruption in terms of chains of transmission and really protect against mucosal infection, most of the evidence currently suggests that to be able to achieve that, not just for COVID-19, but for many other vaccines, there's very strong association with antibody. And it's probably because of transudation of antibody onto the mucosal surfaces. Now, in the context of using a vaccine where the S protein is really the target for much of the antibody-induced immunity, how do you see this sort of a construct where the S protein is still based on a prototype virus, sort of working in a context of where the variant that's currently circulating in South Africa, the B1351 variant, circulates? So do you think that this construct would be still robust in terms of overcoming mutations, especially in relation to protecting against reducing infectiousness and protecting against mild and moderate infection?
Shabir, it's an incredibly important question you just asked, an incredibly complex question. And I'm happy to, if you could see this whiteboard, try and draw an answer to that, if I may.
Yes, please.
So the way we understand and look at whether the cancer or the COVID works and S plus N, and at the cellular level, and if you look at this as ACE2 as a receptor, so the antibodies that are generated would try and block this. The problem, as you're seeing now, is that these antibodies that will occur will be completely mutated. But the way an antibody actually works is it doesn't really kill the virus. It actually kills the cell that is making the virus through an antibody-antigen complex or an antibody T cell, what we call ADCC interaction, or an NK cell antibody generation. So it requires the CD8 T cells for the antibody actually to work. However, what happens if this virus enters into the living cell and it starts to express its proteins on the surface, and its N and S proteins? The ability of CD4 and CD8 T cells not only to recognize that, it is the CD4 T cell that drives the memory B cell, and it's the B cell that drives the antibody. It is the CD4 T cell that drives the memory CD8 killer T cell. So at the end of the day, if you ask what is the key to this whole world, it's the CD4 T cell that will drive the antibodies, that will drive the killer T cell, and will drive for both of them memory B and memory T cells. So what I'm saying here is we've not advanced our enlightenment of the immune system beyond 40 years, where because all we knew was antibodies, we now know with great detail this orchestration of CD4, CD8, B cell, memory B cell, memory T cell, and natural killer cell. This is what we've learned in cancer, because this cell could be a cancer cell. This cell could be an infected cell. An infected cell could be a COVID cell, an influenza cell, a TB cell. And in fact, we've shown sterilizing immunity with H1N1 in 2009 using this technique. So when I talk about constructive disruption, I'm really talking about using today's modernized science with genomics. That's why I'm so proud of South Africa, because we want a leading genomics so that we can actually put the right construct in so that it could actually have these systems. So I hope this little crazy drawing of mine, I don't know how to talk without drawing. So if there are students there, this is the only way for you to understand what I call this orchestration of the entire immune system. So I think it's naive, to be honest, because what's happened is regulatory authorities have used this as what they call correlates of protection. So now you have regulatory authorities doing this. You have the academia that says, we're so used to this is the key, antibodies. If regulatory authorities and academia together say that's how we do it, guess what pharma says? Pharma says then this is how we're going to make vaccines. And then guess what? We'll just make a new one every year. And this is what I talk about stuck in dogma. And we have a vicious cycle because we have academia living in what they're used to, whether it be HIV, etc., with antibody world. Regulatory authorities says we can measure antibodies, so therefore that's what we'll regulate. Pharma says, well, that's easy to make. It's quick and easy. First shot, let's go do that. On the other hand, to measure T cells is difficult. But there's an assay which we did for TB called quantiferon. And TB was affected mainly by T cells. So I think our experience from South Africa, and that's why I am so excited coming into South Africa, because I think we as a country, or you as a country now, and I'm not sure whether I'm American, South African. When I say we, I think of myself still as South African, will have the best chance, I think, in the world to lead the world in infectious diseases and take this approach of orchestrating the entire immune system. So I hope that's an answer to a long-winded answer to a very complicated, good question that you put forth. Yeah.
And that's how I think, but so Patrick, I think you raise another really important issue in terms of how to change the mindset of scientists as well as regulatory authorities, the way we usually think about correlates of protection. And as you're probably aware, the regulatory authorities have proposed a pathway in terms of licensure of the next generation of COVID vaccines, which is really again still very much centered around looking at antibody responses. So how do you expect that to sort of overcome that sort of hurdle in terms of changing that mindset?
Well, I asked the regulatory authorities to look at my NHP data, which not is my NHP data, it's NIAID NHP's data, Battelle, BARDA, warp speed. The non-human primate study, which was done completely independently, if they then compare that NHP data, which unfortunately can't be done side by side, it should have been done. That's how warp speed was planned initially, to take all the non-human primates, do a common challenge. Everybody puts their vaccine in, and you look at the data. What's going to happen next? Obviously, the UK is now thinking of doing their own challenge studies. Is that data shows on its face a complete inhibition from day zero almost, within hours, I would think. We obviously didn't measure it within hours, but the first day, a 2 log and 1 log drop in virus replication in the nose and lungs to undetectable levels. So if that's the case, we would reduce transmission, not disease. So the fact that we have an S in our adeno means that we'll have antibodies to S, but we have potent antibodies to N. And as you begin to see now with South Africa, it's not just the RBD of the S, it's S1, S2, the N-terminal domain, and even N. And the most important, these are Th1 dominant, they're not Th2. And this antigen antibody complex is one of the most important things for T cells. So the idea would be is, would you not want to clear the factory rather than futile just try and block it? I'm struggling and torturing with the question, why? In fact, Shabir, if I may, people have asked me, why has this not been adopted? And I say, I don't know. There is no mystery about the fact that SARS-CoV-1 patients have T cells 17 years later. There's no mystery that there's gamma globulinic patients that have recovered from SARS and only can do that by T cells. There's no mystery that we have zero replication now done by a completely independent organization like the NIH in non-human primate by 10 to the 6th challenge. If you could help me with that question, I'd be grateful, because I don't know how to answer the question why. And my only thought would be, it was easy to measure antibodies. It's not easy to measure T cells. The fact that it's not easy to measure doesn't mean it doesn't work.
Yeah, thanks, Patrick. So Patrick, the first question, in fact, I probably didn't give it a full question. Is there any difference in terms of the timelines that it takes to induce T cell memory responses using a sublingual route compared to an injectable?
That's exactly another good question. I love these questions from South Africa. It's exactly the good question, because the question is, would sublingual give you a different form? It'll give you not only T cells, but IgA. And because it's a different resident dendritic cell under the sublingual compartment versus the intradermal compartment, would it give you systemic immunity alone? And the holy grail would be a sublingual under the tongue and an oral capsule in the mouth and a needleless event. We are testing that in our phase 1s. So we have now close to over 150 patients in phase 1s with maybe 10, 12 different cohorts, where we're looking at sub-Q alone, oral alone, sublingual alone, oral and sublingual combination, sub-Q and sublingual combination. So between South Africa and ourselves, we will come up with what we call is the best recipe. And the idea, as I said, 1 vaccine, 3 routes. And the goal is to give you the maximum protection with the most practical logistics. If it needs a sub-Q, then you do a sub-Q, sublingual, and then you send the patient home or the person home with a bunch of capsules. We don't know the answer to that yet. Unfortunately, we did not have the opportunity in the non-human primate. We asked for 3 groups. They afforded us only 2. So we had to choose sub-Q and oral, but we've not done oral alone or sublingual alone. We're doing that in our phase 1 trials.
Thanks, Patrick. So Patrick, how do you see the future of this vaccine? I think you did allude to it during your presentation. Do you see this as being the prime strategy for vaccination, or do you see this more being the boost to top up the initial spike protein antibody responses that other vaccines could induce? So as an example, do you see this being additive to messenger RNA? And again, that's another question in terms of what the place of this vaccine would be in a country such as the US. So how do you see this? Do you see this as sort of a separate portfolio generation of COVID vaccine, or do you see it being synergistic to what's currently authorized for use?
I actually see it as both. And from a pragmatic perspective, Pfizer, Moderna, AstraZeneca, Johnson & Johnson have invested hundreds of millions of dollars in the manufacturing of finished fill facilities, including with Aspen in South Africa. So therefore, their vaccines as sub-Q, giving an antibody to S, could be supplemented by our vaccine, whether it's sub-Q or oral, and potentially, hopefully, just oral, of an N plus S. So we could serve as a universal vaccine to everybody. In fact, South Africa is doing now its 500,000 rollout of Johnson & Johnson's Ad26 to healthcare workers. But there are also healthcare workers that received the AstraZeneca vaccine. So therefore, should we come in and speak to SAFRA and say, let us be the universal boost to all of these recipients already, and we could create a placebo control trial where the placebo is no boost. And then we measure those patients or these healthcare workers that received our boost, the T cells. And unless there's magic, those that without a boost will not have a T cell to nucleocapsid. We would now generate a T cell to nucleocapsid and to S. I do know that, in fact, we can boost because we've done the recall experiment. And I'm happy to share with you, it's published, where we've taken convalescent patients, taken the T cells in the test tube, made a dendritic cell, vaccinated that with our vaccine, and boosted and proliferated the CD4 and CD8 T cells. So the answer is yes, we could be the universal boost for the world. And yes, we could also be the prime and boost for the world. However, I don't believe any one single company has the capacity to do this for all. So one of the things I'm very interested in doing is coming to South Africa, generating the capacity, not just for South Africa, but self-sufficiency, not for South Africa, but for Africa. Because this idea of vaccine nationalism now is such a short-sighted, wrong-headed way of thinking that we need the entire global community to be protected, and most important, those who can't afford or get access. The way I see it, 1 billion people will not be vaccinated. So if we are just the vaccine for those 1 billion people, I would be happy.
Yeah, Patrick, I mean, I think we're going to loop back to this whole issue. Like you mentioned, the science that's taking place in South Africa around COVID-19 is really phenomenal. And I'm really proud to be part of that. But at the end of the day, it still remains that right now, South Africa, the African continent as a whole, simply hasn't been able to get one vaccine even into preclinical trials, let alone into clinical trials. And this is something we probably want to visit a bit later in terms of what do you think is lacking that despite the caliber of scientists we've got in South Africa and on the continent, we haven't been able to sort of undertake that leap in terms of going into R&D when it comes to vaccines. But before we get to that, so my question to you is that we're talking of this being possibly a boost in the context of another vaccine, but the reality that exists in South Africa based on the latest Zero surveys is that in fact, after the first wave, as many as 30% of South Africans, and in some settings up to 40% of people have been infected with a prototype virus. And after the resurgence, we probably now got more than 50% of the population that's been primed by natural infection. So where do you see the added value of the type of construct you coming where natural infection itself would also induce T-cell immunity? I'm not too sure whether that repertoire of T-cell immunity induced by natural infection would be as robust as is induced by a vaccine. But in a context where there's been such a huge force of infection that's already taken place, but we're still experiencing these resurgences, which include large number of people dying. How do you see the sort of rollout of a construct such as yours contributing?
Shabir, that's exactly why I desperately want to work with the network in South Africa and work with SAFRA. Your natural infection, and what we've now learned about natural infection, is that the natural infection of the virus, it has the capability to have immune evasion. What the virus does when it naturally infects, it actually deacidifies the component in your cell called lysosomes. And by deacidifying it, it prevents that nucleocapsid from being broken down into its tiny pieces. Therefore, the T cell activity and antibody activity is muted and actually not that long. what we have then will done, those are one of the most valuable opportunities for us to come in and actually even prime a previously naturally infected prime or boost, whatever way you want to look at it, with our N+S. Because now we would increase T cell diversity and boost that N. We'd increase T cell diversity to that S. We'd boost those antibodies. In fact, we had one patient in our phase 1 trial that inadvertently must have had an infection but didn't tell us, or he or she didn't know, because post-analysis there was M protein, and our vaccine doesn't bring M protein. That patient had a massive rise in antibodies and gamma interferon within 14 days after our vaccine. So the idea for me to come in now and say, let us approach either the vaccinated healthcare workers or the infected 50% of the population with our safe vaccine, and generate the capacity and manufacturing capacity in South Africa for South Africa, make that a South African vaccine, and for me to come home and do that would be truly sort of a quest. I'll tell you a little story, a personal story of mine, if I may. As you know, I was a Chinese medical student at Wits during the apartheid. Like you, I got into trouble. I was taken to the police station I was pulled aside, and the policeman says, "Where's your ID card?" And my response was, "Where's yours?" Which wasn't too smart a response for a student, but and I was summarily hauled into the police station. And so I understand what we went through. And straight after that, I got appointed to Wits as the first Chinese to ever work in a white hospital. As you know, I graduated in 1975. In 1976, when I was an intern, Soweto had its riots. I then volunteered to go to Baragwanath to treat these children. After my internship, I was offered a full-time job at Wits, at the Johannesburg General Hospital, and I turned it down, wanting to go to the TB clinics in East London. And that's what I did. I realized that there was no technology that I had in my hands other than a streptomycin injection with no x-rays for these children. And I was either hurting them or not helping them, and said I would go away and come back and bring technology to South Africa. I thought it would take me 5 years. I miscalculated. What we have in South Africa is amazing, amazing clinical material of patients that we could help with next generation technology. We have amazing, dedicated scientists. Chris Barnard made breakthroughs, Byberg made breakthroughs, Bothwell's made breakthroughs, Barlow and Seftel, and we can go on. There are giants of medicine and scientists and clinicians in South Africa. We've now extended that by taking on the genomics. What we do not have is the pharmaceutical industry following suit of generating biologics, generating techniques and technology that is innovative, that can actually take and translate things like natural killer cells, things like CAR T cells, things like stem cells, things like next generation vaccines, all biologics that'll change the course of cancer and infectious disease. We developed that now in Los Angeles. My goal is to transfer that completely into the hands of South Africa.
Yeah. So thank you, Patrick. Patrick, so that just reminds me, I'll be held to account by the university if I don't pitch it to you, that we have identified a site for you to set up your manufacturing facility around the corner from the university. So just keep that in mind, which speaks to another issue. In fact, the next question that came up, and obviously it's early days, is what do you see as being the cost of this sort of vaccine relative to what's currently on the market?
I don't know that, and to be honest with you, I, however, do know that if we get to the capsule and tablet form, it would be significantly less, and that's what we must do. We must drive this to an affordable cost, because this vaccine is needed by the poorest of the poor. And so the answer is, I can't give you an honest, truthful answer because we've not scaled. But we do know that people at Serum Institute, for example, say they can make this for $3 to $15 or $3 is what is purported in the public market. But if we can do that at that level, at that scale, that should be the goal.
Yeah. So the next question is, do you foresee any sort of challenges in terms of bioavailability of the oral formulation?
No, because it's not bioavailable in the normal sense of a small molecule. It is actually the virus being alive, meaning it's a non-replicating virus, and surviving the heat, surviving the body temperature, surviving the acid of the stomach, surviving all of that, and then getting into the lower gut. When it gets there, it releases, and there's a thing called the CAR receptor, the coxsackie adeno receptor, that's in your guts and in every cell of your body, and it will bind to that and then get taken up. So the bioavailability is a biological availability. It's very much, it's a viral vector, basically. It's a viral, not safe, non-replicating viral vector that's taking in the COVID sequence.
The next question is something that we've discussed in quite detail previously. In fact, I think when we had our first call, we touched on this issue in terms of using the Ad5 as a vector in the context of a high background seropositivity in the South African population and many of the African populations. Whether that sort of use of that sort of a vector might attenuate the immune responses to the vaccine. And then coupled to that is another question in terms of what is the immunogenicity against the vector itself.
So this is why this is so exciting, right? Because I'm very familiar with the STEP trial and the first generation Merck vaccine trials for HIV. Firstly, the trial itself used the first generation Ad5, where the Ad5 fibers itself created immunogenicity. And that's why you can't do repeated Ad5s. We have modified that Ad5 completely, where the immunogenicity is actually immunologically stealth, so that the fibers do not produce, and shown in humans, many patients with Ad5 antibodies that we can give it repeatedly. I don't know if Phil is still there, but if we could show that one slide that has the Western blot of the Ad5. Phil, if you could do that. The importance of that is that this, therefore, should obviate any of immunogenicity from the vector itself. So here is the first generation. So this is what CanSino uses, Russia uses, and that's why Johnson gone to Ad26, and that's why Chad Ox. This is the fibers that come out of all the first generation Ad5 in the STEP trial. This is what happens with our Ad5, very little, and what we call is immunologically stealth. I think it's so important to create this differentiation. The other element with regard to HIV study, that STEP trial that was done, it was an ad hoc analysis and the science is still murky. Having said that, we've taken every precaution to not only to monitor, not initially to put this into HIV high risk patients. As you know, that was in males mainly, uncircumcised men. So this is where we've taken that approach, and we're very cognizant of this. But we do believe, however, that this is not something we would see with the second generation Ad5.
Thanks, Patrick. So the next question that's been posed, and I think you have covered this, is your view of this really mainly to focus on T cells, or do you still think there's an important role for B cells and antibody-mediated immunity?
It's both. What we've demonstrated is, and in fact, I'm working with an investigator at the University of Alabama, where we'll be measuring the memory B cells, plasma-like memory B cells, which appear within 10 days after the vaccination. So you need to capture it very early. It appears and disappears because it goes into your bone marrow. So, and there are markers to be able to measure what we call these memory B cells. In our non-human primate study, again, if I may show that one slide, Phil, on the memory B cell, just to take you back here to this slide, here you notice there's, during the time of vaccination, when they measured antibodies, the blue is a placebo, the red is our vaccine animals. Here's the vaccine animals. And you would think there's very little antibodies. On the day of vaccination, the placebo had no antibodies, but our antibody, our animals, all 5 of them, shot through the roof. It's literally 1 to 7,000 dilution. This can only be the effect of memory B cells. So here is memory B that's being generated post-vaccine and now shot through the roof. So the combination of memory B cells, antibodies, and T cells all working together resulted in slide 24. Again, just to repeat, resulted in the red of the viruses being completely inhibited. Shabir, I don't think people recognize this important fact. If you look at other NHP data, when they challenge, the viruses grow for a day or two and then come down. You notice there's no drop, zero drop zero growth, but an immediate drop in all compartments, meaning there's resident B cells and resident T cells. Only with resident B cells and T cells could you get this kind of clearance. So when you ask, my trying to fight the dogma of antibodies, which is right here, when you have all of this in your body that you can activate, and when you get a result like that, that is quantified by subgenomic RNA and qPCR, by a body that's completely independent, I don't know what else to say.
Yeah. Well, I can say a little about that. I think one of the interesting results in South Africa is the J&J vaccine, which induces a very similar sort of neutralizing antibody response as the AZ vaccine. And I would expect that in the lab, there would probably be a complete knockout in terms of neutralizing antibody activity against the B1351 variant. But yet we're seeing 89% protection against severe disease. So I think it really speaks to the importance of T-cell immunity probably being the major mediator for severe disease. Just extrapolating from that single vaccine and what we see in terms of neutralizing antibody. So Patrick, the next question, in fact, is from a neighbor down the road from you. We've got a number of people, alumni that have joined the call as well. Thank you very much. Gary Levinson, who I think graduated with you. And the question is, do you think that current vaccines induce sustained cell-mediated immunity?
The truthful answer is no.
Okay. We'll tell. Do you foresee any sort of major issues in terms of side effects using the sublingual or the oral route?
So far, we've not seen any. We've already given participants sublingual. And there's been, and both sub-Q, oral and sublingual, we've seen 0, and I really mean 0, grade 3 and grade 4 side effects. They mainly be in grade 1, grade 2, either local site injection, tiredness, little temperature, which we want, and nothing else.
You don't think that would sort of induce enteric inflammation and cause GIT symptoms?
We've not seen that in these non-human primate studies, which are 10 out of 10, and no health effects whatsoever. And in our mice data, again, no toxicology. And now we're doing the 2-legged mice study. So,
so the next, have you been looking at this technology in the field of breast cancer as a target for genomic therapeutics? Or let me read it properly. If you looked at your technology at breast cancer as a target for genomic therapeutics?
What an excellent question. So yes, we have an adeno HER2 in trials We have an adeno-CEA, adeno-MUC1, adenobrachyury, adeno-PSA. And what's exciting is, and the paper is published, and Phil, if you can go to that one slide with all the papers in cancer, the first slide. Here, I refer you on the right-hand side, the cancer experience with this adeno. Adeno-CEA, we were actually shown not only in phase 1 and phase 2s, we can actually give it repeatedly, safely, but actually prolong survival in third-line colon cancer patients. In this paper here, so this is published, and it's given even in patients with immunity. Then we've taken MUC1 and brachyury. Brachyury is an intracellular protein that's present in many cancers. So brachyury is akin to nucleic acid to us. And this has now been shown by, ironically, just got published in October 2019 by the NIH, that we were able to induce potent CD4 and CD8 T cells to this. Think about this, these are self-antigens. We've then done a neoepitope and shown complete clearance. And then again, this is the NCI. And now we've done PSA and now shown almost a 50 to 100% sometimes reduction in PSA levels of still early trials. So the answer is, this is a vector platform. This is truly a vector platform that has multiple indications. And that old drawing I tried to poorly do to show that we need to activate macrophages, we need to activate CD4 T cells, CD8 T cells, B cells, and antibodies, that's what this thing does. And in the meantime, in 2009, we showed neutralizing protection with H1N1 and published that. And within 8 weeks, we're able to develop a vaccine. And ironically, October 2019, this paper got published on Lassa fever. So these two papers got published just as COVID hit, this paper and this paper.
Yeah. So Patrick, I think your PA told us you've got a hard stop at 7:30, which is really, really unfortunate, because we've got many, many other questions, and I don't want to take advantage of your schedule. So we're sort of reaching the end of this. Apologies to people that I haven't been able to get to your questions. I think they are really good questions. And Patrick, unfortunately, that forces you to have a follow-up webinar once your phase 1 results are ready. And then we can actually look at all of these other questions. Patrick, it's been a real pleasure, but perhaps some parting remarks from you in terms of, why do you think, like I said, the one thing I wanted to look back to was this whole issue about why is it that Africa as a continent, including South Africa, hasn't been able to do this transition when it comes to R&D at a basic science level, and then take biologicals into the clinical trial space. What do you think we're doing? What do you think we're not doing? What are some of the thoughts you could share on that?
Well, I think South Africa has the opportunity to have the human intellect capital. So what's exciting is that South Africa has maintained its lead vis-a-vis Africa, and maybe now even the world, with infectious disease in terms of maintaining its human capital and its universities and its strength. What it's not done, because of the developing world nature, or developed world nature, is actually generated capacity. And that was what I was fearful of many, many, many years ago. I had this conversation with Bill Gates when I was running American Pharmaceutical Partners in the United States, when there were 87 deaths in the United States from heparin. And my company, was one of the largest supply of heparin, safe heparin, for all of the United States. I wanted to bring that capacity into South Africa, and obviously didn't have the time or resources. I now do. I think what is missing is the generation of taking translational science, which we are fantastic at, clinical science, which we are fantastic at, clinical patients, which we have many sadly, that have great need, and creating the gap that is there, which is capacity, the capacity of generating the material so you're self-sufficient, controlling your own supply chain. We have great resources to control your own supply chain. And I think that's what's been missing. And you need people in industry that have understood scale to be able to bring that to the country, and have leadership at the government level and at the provincial level, and now obviously with people like yourself, Shabir, at the academic level, to all partner in this to really make it a sustainable opportunity. And COVID has unearthed this inequities. And now's the time just to be able to solve that, and you can.
Patrick, thanks a lot for your time. There's a number of comments which I haven't been able to get to, many of them forwarding greetings from previous classmates of yours.
I'd love
really
to have a reunion and find out what they're doing, and maybe play them a basketball game if they can play basketball.
Well, yeah, I think someone mentioned that they managed to dig up your photo of being the captain of the basketball team, but which I haven't seen yet, but we certainly will share that to the group. And I'm glad to say that the guys on your side, your team, has agreed to a follow-up webinar without your knowledge, but thanks to them for agreeing to that. Patrick, thanks for your time, and hopefully we can get you to South Africa at least for our centenary celebrations next year. But we certainly will continue our discussions in terms of working with you at multiple levels to see your dream of returning to the country and investing in the academic capital in the country in the near future.
Well,
so thanks for your time. Great pleasure hosting you, and we'll chat soon. And thanks to everyone else for joining.
Well, thank you for wonderful questions, and really look forward to coming home and visit.
Thanks, Patrick. Keep well.
Okay, bye-bye.
Bye.